Hi everyone, today's video is on polymerase chain reactions, or PCR. This is what a PCR looks like, but this picture doesn't do you much good until you know some words. Key terms like amplification and transformation. Amplification is the creation of copies of a specific sequence of genetic code. So, little visual representation. Let's say you have specific strands of DNA, and you want to amplify this sequence. What that means is you want to make copies of it. That's it. And that's actually what PCR does. Now let's talk about transformation. Transformation is this kind of application of amplification. During transformation, genetic manipulation of a cell is done by incorporating foreign genetic material into the native genetic material. So, once again, you have some strands of DNA, and during transformation, a sequence is inserted right in the mix. And oftentimes, that sequence is acquired through amplification. So now you can see how these two are kind of incorporated into this one machine. Okay, so some quick background once more on this genetic material we're talking so much about before we get into PCR. DNA. You've definitely heard it. It's deoxyribonucleic acid, and it's what gives all life. DNA exists as this double helix, which means it looks like two strands kind of rotate around some invisible axis. And if we were to unravel these two strands, they would run in anti-parallel directions, which means one goes from a 5' carbon to a 3' and the other from a 3' carbon to a 5'. And inside, within these two strands, are these nucleotide pairs that are bound. The sequence of these pairs is what gives life, its uniqueness, its diversity. Okay, so let's write this down. This is an important word, nucleotides. And nucleotide pairs come in two forms, A with T or G with C. And they literally run along the entire strand of DNA. The combination and the order of these nucleotide pairs is what gives specific coding its value. Oops, that's supposed to be an A. And it runs in both directions along DNA. Okay, so now that we kind of understand the basic structure of DNA, let's apply it to PCR. PCR. It's a pretty simple process actually. It has 3 steps that repeat 25 to 35 times. The first step is denaturation. The second step is annealing. And the third step is extension. So, like I said, it happens 25 to 30 times. Okay, pretty simple stuff. The first step, like I said, is denaturation. And this occurs once the PCR heats up to about 92 degrees Celsius. Kind of hot. So at this point, denaturation means that the double-stranded helix of DNA separates so that the nucleotides are no longer bonded. And the whole point of PCR is to kind of amplify a specific sequence of DNA. And I'm going to mark that right here. Let's say this is some gene I want to amplify. Now our second step occurs, annealing. Annealing occurs once the PCR cools down a little bit to about 62 degrees Celsius. And during annealing, the gene sequence that we're trying to amplify kind of gets targeted. So, once again, we still have these separated strands, we still have these sequences of genes that we're trying to amplify, but now we add a third component, primers. Primers bind to the beginning and ends of genes. And they're kind of like markers, starting points and ending points for our actual DNA replication. And like I said, because of this anti-parallel direction of the strands, the primers, there are 2 types of primers, our forward primers and our reverse primers, and both are imperative to ensuring that amplification of the whole gene occurs. Now our last step is extension. Extension happens once the PCR is once again heated up, but this time 72 degrees Celsius. We still have our strands of DNA. We still have those genes that we're targeting. We still have those primers, our forward and reverse primers, but now we add another component, polymerase. Without polymerase, this would all be useless. Polymerase is an enzyme that allows for the replication of DNA. So polymerase is going to move along the strand of DNA in both directions. And it's going to bring nucleotides from the surrounding solution to bind with these unbonded strands, unbonded pairs of nucleotides already on the DNA. So once extension finishes, you're going to have something that looks like this. Your original 2 strands that had denatured, and then this new strand of DNA that is bound to it. Because of the starting and ending points of these primers, these sequences are now shorter than the original DNA. And the process continues. These 2 strands of DNA, these 2 pairs of DNA actually, are going to denature, anneal, and extend until thousands and even millions of copies of DNA are at hand. So, what did we do? This is us, our lab group. Our goal was to use PCR to amplify the TEP1 gene. The TEP1 gene would later be transformed with budding yeast, but for now, let's focus on how we did this with TEP1. This was our list of materials. Okay, and we were creating a 50 microliter solution to put into PCR, so these are the numbers we were dealing with. Except we encountered a problem upon seeing all these necessary and required values. Our one biggest issue was equipment limitation. We were going to be using micropipettes to create this solution, and our micropipettes only went down to 2 microliters. But if you look, this value, and this value all required measurements less than 2 microliters. Okay, and just for record keeping, our template DNA was TEP1 at a concentration of 36.9 nanograms per microliter, and we use 2 microliters of that solution. Anyways, back to our problem. We had less than 2 microliters for these 3 components, so we couldn't use our micropipettes for ourselves. It's okay. Our solution was teamwork. I just like that picture, but that has nothing to do with it. Our entire class is made up of 4 groups, each one amplifying a different sequence on DNA, a different gene. If we were to multiply these 3 quantities that we were required by 4, they would all be greater than 2 microliters, which means we could use our pipette. So that's what we did. We created a solution of all the components that were common to all of our groups. So that included, let's just take this away, it's kind of distracting. The components that were common for all of the groups were the buffer. Why is this not working? Oh, here we go. The buffer, the this long substance which actually helps polymerase, the DMSO, and the polymerase itself. These were all common to all of us. And if we multiplied their numbers by 4, they would all be greater than 2 microliters, and we could use our micropipettes. So once we created this one big solution for all of us, we then separated it again amongst us four. And then we added our variable components that included the primers that are specific to each of our genes, the template DNA itself, which is different for each, and the amount of nuclease-free water that would actually reach 50 microliters. Because of the different concentrations of the template DNA, some groups had to add more or less of that DNA, which would in turn affect how much water they would actually need for their solution. But this whole process worked. We created one big solution, we shared it amongst the four groups, and then we added whatever we needed. That solved the problem. Now, did it work, is the question. Well, in short answer, no. Using gel electrophoresis, let's write that down, that's an important process. Gel electrophoresis. The PCR solutions were put into this kind of mechanism that had a negative end and a positive end. And our solutions were put in there. Negative end and a positive end. And like I said before, the backbones of DNA have a negative end. And what happens is once this electrode is turned on, the DNA is supposed to run towards the positive end. And the distance it travels kind of determines the length of the DNA and whether or not our DNA was successful in amplifying the specific gene. As you can see here, this was the control, and we were group 2. We had no movement of our DNA, which suggests, but of course does not completely solidify the fact that our PCR solution may not have completely worked as we had hoped it would. But it's okay, because we can try again, we can try to reduce our human error, we can try to make sure that our gel electrophoresis is working properly, and once it does work properly, we will continue on our process of using PCR for transformation. Thanks, and shout out to Dr. Van Dyken.